At about 4 weeks after infection, the first Cercariae emerge from the snail, and they're soon escaping daily in swarms. They're about 0.5 millimeter long and are actively swimming organisms that travel through the water with characteristic movements, usually tail first. Their life is short, about 48 hours at most, for they have no functional gut, and their rapidly expended energy reserves cannot be replaced. The Cercaria has an elongated body region and a long forked tail. It also has an oral and a ventral sucker by which it adheres to any substrate. The external surface of the Cercaria is covered with spines that are backwardly directed. These help it to penetrate the skin of its next host, since they ensure that the organism can only travel in a forward direction. Each Cercaria has a secretory complex comprising 3 types of unicellular glands which open through the oral sucker. This electron micrograph shows the folded gland openings. The glands contain powerful proteolytic enzymes which help the Cercaria to digest its way through the skin of the host. Here, Cercariae are penetrating the tail skin of a mouse. They attach with their suckers, digest, and wriggle their way through the skin. Once the body is inside, the tail drops off but remains active. The parasite, now called the schistosomulum, is safely within its new host. The sequence is now repeated in diagram. A Cercaria attaches and makes a hole in the epidermis. It deepens the aperture and enters it by elongating its body. Shedding its tail, it migrates through the dermis and fat layer, where it turns, seeking a blood vessel. On finding one, it enters and starts its journey round the body of the host. It's carried in the bloodstream first to the lungs. By this time, it's become longer and considerably more slender, and lost its mid-body spines, although spines are retained at both ends. The spines, together with rhythmic activity, enable it to migrate along the tiny lung capillaries, which are often smaller in diameter than the larva itself. The flukes leave the lungs through the pulmonary veins and pass via the left side of the heart to the liver. The earliest migrants arrive in the liver 6 to 8 days after infection. They become short and squat, begin to feed, and their intestines fill with dark hematine pigment. 2 to 3 week old juvenile flukes are now more recognizable as schistosomes, but they have a convoluted system of folds and ridges on the surface. By week 4, however, these folds are less conspicuous and dome-shaped elevations appear on the dorsal surface of males. They clearly represent early stages in the development of surface tubercles. Male and female flukes pair at around week 5, and the male tubercles take on their characteristic spiny appearance. The paired flukes migrate through the portal vein to the mesenteric veins, and egg-laying begins. That completes the life cycle of the schistosome. We now have some observations on one of the most intriguing features of the life cycle, the transition from free-swimming Cercaria to parasitic schistosomulum. The Cercaria has to adapt very rapidly to its move from fresh water to body fluid, a temperature change of about 20 degrees, and a hostile environment. The most dramatic changes take place at the outer surface of the Cercaria. This is a diagrammatic section through a Cercaria. The surface is spiny and totally covered by a typical three-layered membrane. The outer layer is the glycocalyx. This is an electron micrograph of the three-layered membrane. Under the membrane is the tegument, a syncytium with no cell walls. Its nuclei are located in deeper cell bodies joined to the tegument by narrow, twisted connections. These are layers of muscle cells. This is how the section appears under the electron microscope. When the cercaria enters the tissues of its new host, large numbers of membranous secretion granules are synthesized in the subtegumental cells and pass up into the tegument. The bodies join together and then connect with the surface of the larva to liberate their membranes to the exterior. The original trilaminate membrane and the glycocalyx are cast off in a mass of microvilli. This action not only serves to distract the host's immune system, it also sensitizes the host against subsequent schistosome infection. The parasite rapidly covers itself with a new 7-layered membrane derived from the membranous bodies. The larva further confuses the host by disguising its surface with proteins derived from the host's red blood cells. At an early stage of infection, it's also thought that the host produces a class of antibodies which attach to the invading parasites. Their function is rather puzzling, in that they're not aggressive towards the parasite. Furthermore, they serve to block the killing of the parasite by other toxic antibodies. Later, there's a switch, so that the toxic antibodies predominate. This could explain the finding that young children playing in infected water have no resistance to schistosomiasis and may become heavily infected. But as they get older, they develop an effective immune response that kills newly invading parasites and thus limits the number of schistosomes they carry. Because man eventually develops this partial immunity, concerted efforts are being made in many research laboratories to analyze the mechanisms responsible for killing the flukes and to characterize the parasite antigens against which the host's responses are directed. Present research gives hope that it will be possible to design and produce a vaccine for human use. It would be an acceptable and desirable first step in disease control if the vaccine could reduce the debilitating pathology caused by schistosome eggs. Schistosomes, like many other parasites, have a variety of ways of avoiding the lethal effects of immunity. Shedding of microvilli, using host protein as a disguise, and the development of blocking antibodies. These are but 3 of their many tricks. A really successful vaccine will have to be several jumps ahead of these highly sophisticated evasive strategies. Until that time comes, we must rely on improved standards of hygiene to break the life cycle of the parasite and drugs to reduce the infection. But the problems of expense, the need for repeated drug administration, and the ever-present threat of drug resistance mean that researchers are working against the clock to develop an effective vaccination program.